Thank you. Bit technologically challenged still. I think I would have had it sorted by now. Well, welcome everybody to tonight's Coastal Corals talk. And we will start by an acknowledgement of country. So because we're online, we're all coming to you from different places around the country. And so we all have traditional owners, but we do acknowledge those and all traditional owners past, present and emerging. So tonight's online event is our own awesome Ilha, presenting her research on environmental DNA to monitor benthic communities in Quandamooka. Who is Reef Check Australia? Well, you probably know because you're here, but we're a not-for-profit citizen science organisation, and we believe in protecting reefs and oceans by empowering people, ordinary people like you and I, to get out there and survey reefs and help educate the public. Housekeeping, so some online etiquette, which I think everyone's already doing this anyway. So we like to keep our videos off, just helps with internet speed. Keep your speakers on mute, and please use the chat box. Pop in any chats, any questions you think of as we're going so that you don't forget them, so we can ask Ilha at the end. If you're not on our mailing list to receive our monthly e-news, shoot an email through to me at seqsurveys@reefcheckaustralia.org, and I can put you on that mailing list, and that will let you know what we've been up to and what we've got coming up. As we said, chat box for questions as you go. Group photo, we'll try and remember that if we can do that before I hand over to Ilha. Upcoming events, we've got a few coming up. So this Sunday, for those on the Sunshine Coast, we've got raffles with Your Mates Brewery at Warana. That kicks off at 2 o'clock with music. Raffles kick off at 3. And all proceeds from the raffles go to Reef Check Australia. And you can win some awesome prizes such as slabs of beer, vouchers for food and drink at the at Your Mates, and they have awesome food and beverages. So that's this Sunday. For those up in Townsville, we have the Healthy Waters Arvo, where the Dry Tropics Partnership are releasing their Waterways report card for 2022-23. And we have a pop-up event in the afternoon from 2:30 to 5. We'll be there with a stall, and that's at the Townsville Sailing Club on the Strand. And for those wanting to become an ambassador, we've got ambassador training coming up, commencing on 27 July. Be running online for 3 nights. So that's the 27th of July, 3rd and 10th of August from 6 o'clock. We allow 3 hours, but normally it doesn't take that. And then we'll have an in-person session at some point, depending on your region and the events we've got going on. But you can jump on the website and find out some more information about that. The Gold Coast Whale Festival is on at Burleigh Heads on the 12th of August. Again, we're going to have a team of people down there at that one. And there's lots of awesome events going on that day, including a land-based whale survey. So jump on to the Gold Coast Whale Festival website and you can get a full list of events and pop in and say hi to our team. For those in Caloundra or that area, we have the Plastic Free Sea Festival in Happy Valley on Saturday, the 23rd of September. That is, it runs from 8 till 11, run by Visionary Ocean Warriors. And then, of course, Caloundra Music Festival is on at the end of September to early October. Tickets are now on sale, so jump onto the Caloundra Music Festival website to grab your tickets for that one. And then next month's online event, this is awesome. We've got a PJ talking about her research into tiger sharks and understanding their movement along Australia's east coast. So that should be exciting, and we'll post up some more details about that one soon. And if you'd like to get involved, you can volunteer. So we have 2 ways. One is to become an ambassador, and the other is to become a survey diver. You can find out all of the information on any prerequisites on our website. And as I said, we've got an ambassador course coming up at the end of July. And don't forget to follow us on all the socials and stay tuned with what's coming up. And then we have to thank all of our sponsors who make events like this possible, including Townsville Council, Sunshine Coast Council, City of Gold Coast, Port of Brisbane, and the Great Barrier Reef Foundation. And then a thank you to the team behind the scenes, which is Ilha and myself and Jodie and a couple of other people whose photos I don't have up there yet. But thank you. Without them, it can't happen. And tonight's speaker, well, you would have got our note or a message and said, yeah, Ilha's, well, she's one of our awesome team leaders, but she grew up in East Africa, done a lot of work in research assisting and a lot of study, and is currently working on a PhD. So good on her, but she's going to chat for us tonight. And if this is, I will stop sharing now.
Really?
Okay. So does everybody want to turn their videos on and we'll take a screenshot? No? Oh. Oh, hey Sophie.
Good evening.
Hi, Raz. Okay. Let's see how we do this. As I said, technologically challenged.
Don't worry, I am too this evening.
Thank you very much. So we can turn them back off again and you can share the screen.
Awesome. Can everyone see that?
Yeah.
Awesome. Thanks, Julie, for the introduction. So I'll get right to it. So this talk is really going to be talking about monitoring benthic communities in Quandamuka or Moreton Bay. And I just wanted to preface the talk by saying that this was a really collaborative endeavour, and in particular was led, or at least the aspects that I'll be talking about were led by a team based at the University of Queensland in Brisbane. So the way I'll structure the talk is just give you an overview of the project and our work in Moreton Bay. I'll then and that'll include a bit of a talk about the aspect of the project I've been working on and our methods. And then I'll lead into talking a little bit about eDNA or environmental DNA monitoring, mainly because this is something I've had a lot of people approach me about over the last couple of months, and it's something that's gaining a lot of interest. So I thought I'd give a bit of an overview of the method and how it's used nowadays, particularly as a primer for something that I'm developing with a few colleagues, which is to develop some sort of workshop that we're hoping to present later in the year, which will hopefully be something open source that the public can engage with. And it'll be an eDNA, I guess, package or overview of how you can use it in monitoring and conservation. And so from there, I'll talk a little bit about our preliminary environmental DNA findings, and then also the next steps for this project, given that it's ongoing. So for part one, we'll get started. And I just wanted to, I guess, describe the area that we're working on for anyone that maybe is not familiar. So we work in Moreton Bay, which is just off the coast of Brisbane. And it's an area that's frequented by a lot of people for a number of reasons, whether that's recreational fishing or simply holidaying on some of the islands. And of course, it's an area of a lot of commercial and industrial use. And it's also in close proximity to the Port of Brisbane and to the mouth of the Brisbane River. So I'm sure a lot of the people here tonight will already be aware that that comes with a lot of issues and strain on the ecosystems and organisms that inhabit that area, given its proximity to a city like Brisbane. And what is perhaps less well known is the fact that this region actually harbors a lot of coral communities, which is less well known. And of course, they're different to maybe what you might imagine on the Great Barrier Reef or in places like Indonesia, but they are nonetheless coral communities that are unique and diverse in their own right. And this is, I suppose, the aspect of the project that excited me the most. And I'll be talking a bit about it later today. And so our concerns for the bay as a whole, and also in particular the coral and benthic communities, led to the establishment of this Sustainable Urban Seascapes Moreton Bay project, which is a huge collaborative effort. And it was really born out of the University of Queensland across many interdisciplinary teams from social sciences to ecology and engineering, as well as conservationists and policymakers. And over the last couple of years, we've really tried to engage with stakeholders and the community to see if we can, I guess, lengthen the lifespan of this project and see where it could potentially go in the future. So as a disclaimer, this project was really run over an 18-month period of 2021 and 2020. And that's when it was really funded. And the idea was to use that period of time to collect as much data as possible. And so essentially the project is, I guess, over, we would say, since December 2022, but most of the teams are still, such as ourselves, the team that I work for, we're still very much working on the data, generating that information, and still engaging with the relevant stakeholders so that we can really put a lot of that information into practice. So it is still ongoing. And the project as a whole is made up of 3 sub-programs. So the first one being centered around ecosystem dynamics and biodiversity. The second looking at predicting environmental futures. And the third at sustainable management. And I'll really focus on this first program, the ecosystem dynamics and biodiversity, since that's the one that I've been working in. And what we're really interested in is characterizing and monitoring ecosystems, as well as better understanding the environmental and seasonal drivers of change. So to do this, we decided to monitor a number of sites across the bay, 3 of which are located outside of the bay, including Flinders Reef, Shag Rock, and Flat Rock. So for anyone that doesn't know the structure of the bay, I've put a map for you. So the dots that are located off the 2 separated islands, those are our outer bay sites, And then we have 5 main sites located inside the bay, as well as some samples that were donated to us from some collaborators near the Port of Brisbane. And so as best as we could, given weather and other constraints, we aimed to sample these sites every 3 months over that intense 18-month period of the project, with the intent being to capture any seasonal changes and variations. And so for anyone that isn't familiar with this region or hasn't been underwater in the bay, you can already probably imagine that your inner bay sites, for example, this is a photo taken at Goat Island, which is located inside the bay, can be very different from those located outside the bay, such as Flinders Reef. So your inner bay sites tend to be extremely shallow, sometimes even just 2 meters deep. They're much more turbid and dominated by turf algae, and are very much influenced by any activity that's going on close to shore. So for example, at the mouth of the Brisbane River. Whereas your outer bay sites, such as Flinders, tend to be a lot deeper, they're serviced by oceanic currents, and so they're much clearer and tend to be host to a lot more organisms, particularly larger pelagic fish, and also tend to be covered in a lot higher coral cover, as you can see in the image on the right. And so for this ecological part of the project, we decided to focus on a variety of different things so that we could try and capture the biodiversity and the dynamics that are ongoing at these sites. So the first one was photomosaics. So what that entails is essentially taking thousands of photos across transects at each site so that we could develop 3D and 2D maps of each site. And so that can give you a lot of really interesting information about community composition and cover. And so that work is ongoing and is primarily led by Dr. Gal Ayal. The second part was to look at environmental conditions to, I guess, support some of the findings that we might see in our samples or in those photo mosaics. So we were really interested in temperature and turbidity in particular. So we had some loggers deployed at all those sites. We also had a team that were looking at fluorescence in coral using a diving pan. And then the part that I'll really focus on is the biological samples that we collected. I just wanted to briefly mention that I won't really be talking about the first three aspects of the project, but if anyone is interested to find out more about how that work is going and where it's leading, I'd be more than happy to talk about that. And hopefully some of the papers and articles associated with that work will come out relatively soon. But the reason I'll be focusing on the biological samples is because that's what we use for our environmental DNA monitoring. So for the purpose of this project, we decided to focus on settlement tiles. So you can see them on the far left photo. We also wanted to look at sediment cores. So in the middle picture there, you can see Gull is closing up a small, I guess, 0.5 meter to 1 meter long sediment core. And then on the far right, you can see some coral samples that we collected. Now, the coral samples might not strike you as environmental samples, and I suppose they're not, but the reason I included them was because there's a sub-team within ours that will be looking at the symbiont and bacterial communities that are present in those corals. So in a way, they are looking at the environment or the host and its microbiota. I won't be focusing too much on that coral work since it's still ongoing and something that's being led by a PhD student. And I'll primarily focus on the first two sample types. And so you might be wondering why we would collect these particular samples and why perhaps we might not include something like water, given that water is the most commonly collected, I would say, environmental sample in the marine sciences. Now, the reason being that we simply had a lot of issues with equipment to filter the water samples. And so it was more of an equipment and accidental reason that we didn't include water samples, unfortunately. There are also a lot of other downstream issues with processing DNA that's captured from water, but it is something we'll hopefully include in any future iterations of this project. But we decided to focus on the settlement tiles and the sediment because they actually represent the communities and organisms that are more likely to be, I guess, present for a longer period of time in those particular sites, given that the settlement tiles would represent organisms that settle and recruit into your site, and the sediment samples are more representative of the benthic organisms that are present. Whereas water samples are more likely used in studies that are interested, for example, in fishes. I know that they're used a lot, for example, to look at large predators such as sharks or whale sharks, and they're much more commonly used to look at any organisms that are passing through that environment or that site. So this leads me to part 2 of the talk, which is to, I guess, give you an overview and a brief understanding of environmental DNA, the what, the why, and the how. So for anyone that doesn't know what environmental DNA is or what we're talking about when we say eDNA, it's essentially DNA that is present in your environment. So common examples are water and sediment. And more recently, a mind-blowing sort of research program that started in Europe is to actually look at DNA that's present in air samples. So they use filters to filter out organisms and DNA that's present in the air, which I can't even begin to imagine how that works in terms of, contamination. But anyway, we'll focus on water and sediment for now. And so the reason you might look at environmental DNA is because it allows you to detect cryptic or microscopic organisms. So particularly in marine environments or aquatic ecosystems, it can be really useful, for example, when you're looking at organisms and animals that are really difficult to detect or find, and also microscopic things. So for example, larvae, or in our particular case, if we're looking at bacteria or symbionts, it's just something that's pretty impossible to detect and to identify based on, morphology, eyesight, or any of your more traditional techniques of biomonitoring. And this method is increasingly used in monitoring and conservation, which is why I really wanted to dedicate an entire part of this talk to giving you an overview of it. So I also wanted to highlight that eDNA and eDNA metabarcoding are two slightly different things. So when we're talking about environmental DNA, we're talking about the DNA that's present in the environment. Whereas what we're talking about when we say eDNA metabarcoding is the method or the tool that is used to process environmental DNA. And metabarcoding is just one method that's used. I just wanted to clarify that. And it's the method that's perhaps most commonly used, and at least it's the method that we use. So I just wanted to clarify that. And what we mean by environmental DNA metabarcoding is essentially it's a tool that allows you to simultaneously identify many, many taxa within a given biological sample. So if you imagine a single sediment sample, there might be hundreds to thousands of different eukaryote or prokaryote organisms within that sample. And so metabarcoding allows you to identify as many as possible that are present. And I don't say all, I just say as many as possible, because of course, many of these methods are restricted by, for example, reference databases or the quality and quantity of the DNA that you extract, all of these very fine scale and numerous different things that can really affect your outcomes. And I won't delve into them too much tonight, but if anyone is interested in talking about those things, they're I guess, restraints to the method that we'll be talking about in the workshop that we host later this year, hopefully. And so with this method of eDNA metabarcoding, what you're essentially doing is taking the DNA that you extract from your sample, you amplify it, and you essentially give the DNA a barcode or a tag that allows you to associate it back to its sample of origin. So if you think of a barcode in a supermarket, relate it to the same thing. So essentially you're giving all of the DNA that relates to sample A a unique barcode, and all of the DNA that relates to sample B a separate barcode. And that allows you to differentiate the two different samples. So I'm just keeping it very simple for tonight, and hopefully that makes sense. And so what's happening with all of the technological advancements that we're having, particularly in the genetic field, is that we're increasingly able to sequence hundreds to thousands of samples at a much reduced cost. And because you've barcoded each sample, you can sequence them simultaneously, which makes this method very valuable. And so it has a lot of applications, many of which I've already mentioned, but essentially it makes it a lot quicker and efficient to identify numerous taxa within a sample. I say relatively quickly because, of course, when you're the one doing all the lab work and working through the entire pipeline, it doesn't seem that quick. But if you compare it, for example, to staring down a microscope for hundreds to thousands of hours, it most likely is a lot faster. It can, as I mentioned previously, target organisms that cannot be identified morphologically, like larvae and plankton and microbes. And it's most commonly used to characterize biodiversity. So, for example, if you're interested in presence or absence and community composition. And it's also used for general monitoring over space and time. So, for example, if you're looking at spatial or temporal variation. And it's increasingly used to look at specific target taxa. So a prime example here in Australia and on the Great Barrier Reef in particular is the use of eDNA to target and identify crown of thorns starfish. And so this method and tool can be used across many different organisms. It can be used in a metabarcoding sense where you're looking at whole community composition, or it can be used with more specific methods that aim to look at just a single, say, pest organism or whatever it might be. Sorry, I've just realized that there's been an issue with my share screen. Can everyone still see the slides?
No, they've disappeared.
Yeah, how's that?
That's better. You're back.
Okay, awesome, great. So what I'll do now is just step you through an overview of the environmental DNA process. This is very brief and just an overview, but I thought it would give you a better understanding of some of the data that I'm about to show you. So the first step is essentially to extract the DNA from your sample. In our case, it would be the sediment or settlement tiles. And then you amplify that DNA using markers of interest. And what I mean there is that you're interested in amplifying a particular gene. And so when it comes to environmental DNA metabarcoding, when you're looking at general biodiversity, you're often using markers such as the CO1 gene or the 18S gene. When it comes to bacteria, a really common one is the 16S gene. But I won't get into those types of fine scale details because it can be really confusing and overwhelming. But once you amplify that DNA, so you've got a stretch of DNA that can be, variable in length, you will then sequence that information and then assign taxonomic information to those sequences, which allows you to come to the end of a project and say, sample A has these individuals in it, sample B has these, and make those comparisons and inferences. So this leads me to part 3, where I'll show some of our very preliminary results, in particular looking at bacterial communities, just to give you a taste of what this kind of environmental DNA metabarcoding process can lead to. So what I'll show you in a moment is some of the work we've been doing to characterize bacterial communities in our sediment samples. And we did this by using 16S metabarcoding. And what I'll show is just one time point. So that's the autumn or March, April 2022 samples. And just for clarification, we were only interested in the DNA that we extracted from the top layer of that sample. In other studies, you might look at different layers within a given sample, but we only collected really short, small cores, not long, sort of ancient DNA type cores. And so you might be wondering why we're interested in bacterial communities from the outset. And that is simply because the composition and diversity of bacteria in different samples can tell you a lot about ecosystem health and function, given that bacteria have numerous roles in many different biological processes, such as, respiration, for example, or waste breakdown. And this is definitely not my area of expertise. And so it's something that we are collaborating with the Australian Centre of Ecogenomics, because there are many researchers there who focus entirely on bacteria. And so they can speak a lot more to what these results mean. So what I'll show here is the results for those autumn sediment samples. So what you'll see on the y-axis is relative abundance as a proportion. And then on your x-axis, you have your different sites. And so each bar represents a sample within each site. And then you'll see that there are different colors within that stacked bar plot, and those colors represent different orders of bacteria. And we're just focusing on the most commonly occurring ones. Sorry. The plot can be a little bit overwhelming. And so I just wanted you to focus on the Port of Brisbane and the MYO or Mayora samples. And the reason I separate them out is because they appear quite different in their composition to many of the other sites. And so from the get-go, this is something we were really interested in. And so when it comes to analyzing some of the other samples that we're currently working on, it'll be really interesting to delve into some of those differences and perhaps combining it with some of that environmental information that we collected that I mentioned. So for example, temperature or turbidity, to better understand what might be driving those differences across sites. Another way we can represent the data is to show it as a PC or an MDS plot. And so what this is simply doing is separating out your sites in space based on their differences in community composition. And all I want you to take away from this plot is how you can already see some separations across sites. So if you look at the middle of the plot, there's a lot of different sites that are clustering together, and those are mainly your inner bay sites. Whereas these sites that are clustering as separate clusters, so you've got your grey Flinders Reef cluster, you've got your brown gold flat rock cluster, they are both outer bay sites that are that appear to be quite different in their composition to your inner bay sites, which again is an interesting preliminary finding because it already shows some contrast between your inner and outer bay sites. Another interesting point is you can see your Port of Brisbane in your turquoise color is also a separate cluster. And this is something we're finding in some of the rest of the data set that we're currently working on, is those Port of Brisbane samples seem quite distinct from the rest of the bay. And I don't find this necessarily super surprising, given that the samples were collected right next to the port, where you got a lot of industrial and commercial activity. You've got a lot of products that are coming out from the Brisbane River and influencing the communities that might be present there. And so it's going to be really interesting to have a look in particular at those Port of Brisbane sites. We also did a quick analysis of the alpha diversity measures across those sites. And again, all I want you to take away is just that we're already seeing, in particular, that Port of Brisbane and Mayora, again, the alpha diversity is separating out as distinct in comparison to some of the other sites that we analysed. I also wanted to briefly touch on the invertebrate communities, because this is something that perhaps I'm more interested in, or it's the work that I'm working on in particular, but we're still currently processing a lot of that environmental DNA data. But I wanted to show the photos of some of the tiles we've been working on, just to highlight that even just morphologically at a first glance, we can already see some quite clear distinctions across, in particular, the inner and outer bay comparisons. So if you look at the top two tiles, they were collected at Flinders Reef, which is located outside of the bay, and they were retrieved in March of 2022. And they're really characterized by higher calcareous algae. We also found a lot more coral recruits on those, and in general, just more invertebrates, such as sponges and corals, as well as ascidians and bryozoans were settling on those samples. Those tiles. Whereas if you look at the bottom two tiles that were collected at Green Island, which is located inside the bay, they were really characterized by greater settlement by turf algae and barnacles, and much lower diversity in general of invertebrate taxa. And so there's more to come on this work, and it is really exciting to already find some of these differences. So this leads me to part 4, which is to talk about the outcomes and next steps of this work. And so, of course, the talk was quite data poor, since I only showed you just some of our preliminary data, but what I did want to share is that we're already seeing some clear spatial differentiation as we start analyzing more samples, more sites, more time points, particularly when it comes to looking at our eukaryotes. So, for example, our invertebrates in particular, we're seeing some clear differentiation through space, so across our different sites. We're also seeing clear distinctions between our different sample mediums. So, for example, our settlement tiles are characterizing different individuals and organisms to what we're finding in our sediment cores. And this is not surprising, of course, at all. And it just reinforces why we decided to collect different sample media, so that we could capture as much of that biodiversity as possible. We're also starting to pick up on some clear differences in seasonality, so in diversity and composition across your different seasons. And it'll be really interesting to look into that and see how, for example, temperature might be influencing and driving those differences. Another aspect of this project will be to look at any pre and post flood signals that we might be able to capture, given that, for better or for worse, this project happened to fall across the floods that occurred in Brisbane in early 2022. And so we did manage to capture some samples just before and just after the floods. So we're hoping to look at any signals and changes or shifts in biodiversity in those samples. And so you might be wondering what's next, or, why is this information important, and what could it lead to? Well, what we're hoping is that we can make a better case for why continued seasonal monitoring might be important in the Bay, particularly if more groups, or for example, the government is more interested in looking at restoration or conservation initiatives. It can be really important to use monitoring as a tool to detect and ensure that those policies and those efforts are working in your best interest. So it can be really complementary to some more applied practices. We're also hoping to integrate the data across all of the different programs. So you might recall that earlier I mentioned that we had, social scientists and engineers involved in this project that were all working on very different things. But the big challenge, but something that will be really interesting, will be to marry all those different programs and projects together to create a really holistic understanding of what's happening in the Bay. And of course, what I would argue is a really important aspect of this project is the engagement with stakeholders and the community. So from the outset of this project, we've engaged a lot with different stakeholders to see what they wanted to come out of this project. And it's something that I'm hoping we'll continue to do as the data comes out of this project, because that's really the only way that we'll be able to use this information for policy and practice, which is ultimately why most of us are doing this work. And so with that, I just wanted to thank many of the people that were involved in this project, because it really did take a village, and there were so many different team members involved in this work. So I particularly wanted to highlight Gal, Nick, and Natalie, who assisted with a lot of the field work, and they're also the individuals who were really leading the ecological aspects of this project and the photogrammetry or photomosaic work. So if you do want to follow up on any of that work, just search up their names, and you might be able to find some of the papers that they're soon to publish. And I also really wanted to highlight Karen, Eva, and Cynthia, who helped immensely with the eDNA work and who have really led a lot of this. So my great thanks to all of them. And with that, I just wanted to leave this QR code that shows, it just sends you to a link that leads to the website for the Sustainable Urban Seascapes project. So you can find out information about the different programs, the different principal investigators, and also their contact details. And then, of course, I'm happy to take questions, but you're welcome to email me as well. So thank you.
Awesome. Thanks, Ilya. That was amazing. A lot of work.
Thank you.
Okay, so far the only question we've got is the shipwreck in your first photo. I think that was the platypus wreck, was it, at Peel Island?
Yes.
Yeah. Okay. Sophie. Okay, so does anyone have any questions they'd like to ask, or you've just baffled them all with brilliance?
Sorry, a lot of information. But yeah, feel free to, I think Sophie messaged me, feel free to go to the website of the project. As I mentioned, the funding part of the project is essentially over, but as you can imagine with research, a lot of the work is still very much ongoing, and papers are still yet to be published. And I'm assuming that a lot of the different team members will be speaking about some of their results at upcoming conferences later in the year. I know a few people just presented at AMSA.
I had the pleasure of chairing a few days at AMSA. I was a technology assistant.
Yeah, awesome. Yeah, so I think there were a few people that presented, and
it was amazing because I'm a second year undergrad. It was a fantastic opportunity.
Yeah.
In regards to the data at the port, do you think that the dredging would be having any impact on that as well, Ilya?
I think definitely. So with the port samples, we've only looked at bacteria at the moment. We're still waiting on the sequencing information for the invertebrate communities. But given that it's not like that area is really home to a lot of corals or sponges or anything, I think a lot of our questions and answers will come from the bacterial communities. And so there's quite a few different teams that are working on microplastics and microbes in the bay. They'll be helping us a lot with that work. But I think anything like dredging or flooding or severe changes in temperature or turbidity can lead to some really interesting changes. So our colleagues at engineering have looked at the seagrass and some of the more physical properties of the sediment. So they've done like tests looking at heavy metal concentrations, and they're finding some really scary things. For example, like legacy concentrations of DDT and other chemicals that have been banned for many years, but have still remained present and active in those environments.
Yeah.
Sorry, Julie, it's Raylene. Hey, how you going?
Yes.
The dredging would only affect your turbidity very shortly, depending on your plumes, and most dredging permits would have restrictions on the plumes. So I could see the biggest impact you're going to have is actually in the water quality and that turbidity impact while the dredging is happening.
Yeah, I was thinking whether it would take away, because sort of Ilya's only looking at essentially current eDNA, like whether taking off that top layer of dredging is then putting you into like historical deposits of DNA.
Yeah, you said it's seasonal though, Ilya, didn't you?
Yeah, so we've been collecting seasonally to account for that kind of variation. But you did mention historical, and that's a really good point. So we collaborated with a team at engineering who were fortunate enough, because it's not an easy endeavor, to core and drill down a 4-meter long core. I can't remember off the top of my head where exactly they collected it, but they are using that core to look at historical changes in those physical properties, so things like chemicals and metals. And we were fortunate to grab samples along different layers of the sediment, of the core. And the intention is to look at the historical changes in the DNA. The main, I guess, limiting factor at the moment is just finding a student or someone that would be willing to work on that. And of course, there's a lot of resources that are needed. The other thing is that what we call ancient DNA or historical DNA can be quite tricky to work with. So Karen, who I mentioned as someone who helped a lot on this project, her entire PhD was devoted to ancient DNA, and it can be very difficult to work with given on how things have been preserved, but it is something really, really interesting. And I hope they find the funding and someone interested to take on that project.
That would be really fascinating, Ilya, especially if you start thinking about long-term monitoring with this type of thing, because that's definitely what we've been looking at with Jonathan Weary and eDNA with jellyfish. And looking at the change in communities within the jellyfish over a long-term monitoring, just to try and see if we're going to have that influx of the northern species that everyone's so worried about hitting the Sunshine Coast. Which we've had no evidence. 5 years, no evidence. But we've started providing samples to Jonathan to add into his eDNA database for that.
Awesome. Yeah.
But I can see aspects of what you're talking about for long-term monitoring and climate change, particularly if you've got those historical cores, that will be so valuable. I really hope you find a student for that. That'd be amazing.
Yeah, I hope so too.
Very exciting.
yeah. And also, if anyone is working sort of in the research space and is interested, we've been collaborating with so many people because it took so much to get these samples, and there's so much more that can be done. So if anyone is interested, reach out, because we had a group, for example, Helen Bostock at UQ and some of her collaborators at Griffith are interested in microplastics. So with our cores, we only use 1/2. So they've taken the other half and we'll look at microplastics at the same site. So there's so much more that can be done with a lot of these samples, which is, again, really exciting that we can work together.
Yeah, that's really exciting. Great work.
Yeah, thanks.
Did we have any other questions? Any comments? No? No, that's very good. Well, I'd like to thank you all for coming along. And if you want to run past any of this again, it will be up on YouTube in a day or so. Under the Reef Check Australia YouTube channel, you'll be able to find us. And and reach out if you've got any questions, and we can pass them on to Ilya as well. So thank you all very much for coming.
Awesome. Thanks, guys.
Thank you so much.
Bye.